Good evening. I'm hoping to do this lecture today, despite the snow day, by video. That way you can have your reading day to actually study for any final exams you might have that first Tuesday. So today we're going to be talking about water bonding and polymers. A couple of things that we're going to go over is some basics of bonding that you may have learned in a chemistry or biochemistry class before, the effects that water can have, especially on proteins, and how enthalpy, entropy, and the Gibbs energy are all related to equilibrium. We're going to talk a little bit about pH and buffers that are so important in biological systems, and then we're going to talk about the major biological polymers, and we're going to spend the most time on proteins, but we're going to talk a little bit about lipids as well. With proteins, we're going to talk about the pH dependence of their shape, the structure and function relationship, and a couple of things that can be done to proteins once they're made, and then a very brief little section on enzyme kinetics. So the first thing I want to talk about is the bonding between atoms. There's different types of bonds. Ionic and covalent are the two that you are probably most familiar with. Ionic being an electrostatic attraction between a positive cation and a negative anion. These bonds are really strong electrostatic bonds, but they're not very good for storing energy because in the presence of water, they're just going to be pulled apart by their charges. Covalent bonds are also very strong, and these are the ones we talk about most often in biological systems. Covalent bonds form when two atoms are attracted to the same electrons and end up sharing them with each other, and covalent bonds are how we store most energy. Covalent bonds are either polar or nonpolar. Polar bonds have an uneven electron distribution, so the electrons spend a lot more time around one atom than the other, so you sort of have a positive side and a negative side. Water is a really good example of a polar molecule. There's also nonpolar molecules. For these, the electrons are evenly distributed around the molecule, and there are no charges that you can tell just by sort of holding a magnet up to them. A good example of this would be most of your carbohydrates or fats. Okay. Some other types of bonds you might have heard of are hydrogen bonding and van der Waals bonds. These are intermolecular forces, so they're not as strong as the intramolecular forces. The covalent bonding and the ionic bonding are much stronger than hydrogen bonds or van der Waals forces. However, hydrogen bonding is the strongest of these sort of weak interactions. But it's really important that you understand that hydrogen bonding is really weak, but there are so many of them in a molecule as big as a biological molecule that they really add up. Hydrogen bonding can only occur when a hydrogen is directly attached to a very electronegative element. Usually we're talking about fluorine, chlorine, and oxygen here. Van der Waals forces exist in pretty much every molecule. These are the weakest type of interaction, but again, when you have really large molecules like starches and proteins and fats, you have so many hydrogens floating around on the outside that this very weak interaction can actually add up and become an overwhelming force in holding a molecule together or in its separating. Okay. Water is very important. As you're probably aware, the human body is about 70% water. So whenever we're talking about reactions happening in the human body, we're talking about water. So it's really important to understand how water is going to interact with our proteins and our fats and our starches. So the first thing to understand is that water is polar. The oxygen molecule has a slight negative charge, and the two hydrogen atoms have a slightly positive charge. You might remember water as looking kind of like Mickey Mouse ears, with the two small hydrogens on top being the positive area. Right? Because water is polar, it tends to interact strongly with other polar molecules. So when you have polar molecules, we call them hydrophilic. It's Greek for water-loving. What it really means is that the molecule adheres to water at least as much as it adheres to itself. Often it actually adheres to water more than it adheres to itself. Hydrophobic, the water-fearing molecules that you talked about back in even high school biology, those are your nonpolar molecules. And what happens is the interaction within the molecule is mostly van der Waals forces. And so those molecules are expecting van der Waals forces. So they bond much better to each other to other things that are held together by van der Waals forces than they do to something like polar, which is a little more electrical. And those electrostatic interactions with the charges are really unexpected. And so the hydrophobic molecules cohere with itself much more than it adheres to the water molecules surrounding it. With protein folding, proteins have charged areas and they have neutral areas. So you actually have both hydrophilic and hydrophobic interactions happening. And so you have a combination of these that actually dictate the protein folding or the conformation of a protein. There are also a handful of molecules that are something called amphiphilic. Amphiphilic have two ends, a hydrophilic end and a hydrophobic end. Mostly we're talking about things like phospholipids, where you have a lipid, which is a really long hydrophobic string, and then you put a charged particle right on the end of it. These things often form micelles or membranes. You actually can see an example of this if you have water and oil in the sink. What you'll find is the oil beads up because it's hydrophilic, it wants to stick to itself. But if you add soap to it, the soap has a charged end that sticks into the water, and it has an uncharged end that's kind of fatty that sticks to the oil. And so it helps the oil to sort of get mixed in better with the water. But if you just have water and you put in soap, you'll find the soap makes these little bubbles inside the water. Those are called micelles. It's the same way that a phospholipid forms the membrane for our cells, where you have the phosphate groups on the outside and inside exposed to all the water, and the lipid tails facing the inside, forming the middle layer of the cell. pH is something that we talk about a lot. pH is actually an effect of water. So what happens is water, as it turns out, breaks apart. It dissociates into hydronium ions and hydroxide ions. So if you have two water molecules, it's going to break apart. The hydrogen from one of the water molecules is going to stick to the oxygen on the other one, and you get what are called hydronium ions and hydroxide ions. And this happens naturally. Water has a dissociation constant that actually you already know the dissociation constant, it's 14. We're going to talk about that in a couple of minutes. But pH actually is an effect of water. If you add certain things to water, you can make it form more hydronium ions, or you can make it form more hydroxide ions. It depends on what it is. Basically what happens is if you have an acid, it has a hydrogen that can come off of the acid molecule and add to the water to make hydronium ions without actually making any hydroxide ions. With bases, you sort of have the opposite effect. It pulls the hydrogen off and makes it stick to the molecule, and then you have the hydroxide molecules left in solution. pH is actually a mathematical function. This lowercase p is a math function that means take the negative log of something. In pH, what we're taking the negative log of is the concentration of hydronium ions. These square brackets refer to a concentration. Okay, so a little quick thing about thermodynamics before we go back into pH and equilibrium. Thermodynamics is really about enthalpy and entropy and how they interact with each other. It's about the energy involved in a system. So enthalpy, which we use the symbol delta H. Oops, let me go back. Sorry, enthalpy, we use the symbol delta H. It's sort of a stored energy, it's internal energy. And in a spontaneous reaction, you want the stored energy to be minimized. You want to release whatever energy you can. Exothermic reactions, those are ones where energy is released. An example that is relevant for us is ATP becoming ADP. The sign of the enthalpy is negative. That means that the energy gets released. It comes out of the molecules. An endothermic reaction, that's one where the energy is absorbed. So the opposite, ADP uses energy to become ATP, and there you have the sign being positive. So spontaneous reactions, you want to minimize this internal energy or this enthalpy. So for a spontaneous reaction, you're expecting it to be exothermic with a negative sign for the change in enthalpy. Entropy is sort of another factor that tends to fight against the enthalpy a little bit. Entropy is about disorder or degrees of freedom. It's basically how many different ways can we rearrange the pieces and it's still the same thing. Entropy has the symbol delta S, and it's the energy that we can use to do work. We always will maximize entropy. So the sign for delta S should be positive. If you think about it, right now outside we have snow, a frozen form of precipitation. The water molecules are all locked into a crystal. They really can't move around much. But over time, the snow is going to melt. And when it melts, right, we take even the past couple of days, it's been below freezing, but the sun's been shining. And all that energy from the sunlight allows the water molecules to sort of free up from their crystal form a little bit. And that lets them sort of spread out and have different conformations. It changes the shape of the snow. It melts the snow. So that's actually a case where it's degrees of freedom that's actually winning out over internal energy. Okay. Entropy and entropy tend to compete with each other. And so what we need to determine if something is going to be spontaneous, if it's just going to happen all by itself, is the Gibbs energy. Gibbs energy lets us sort of compare the effects of enthalpy and entropy. So the equation for it is delta G, that's a Gibbs energy, is the enthalpy minus the temperature times the entropy. Temperature is always in Kelvin when we're talking about thermodynamics, so it's on the absolute scale. So basically, if the enthalpy and the entropy are going in opposite directions, so the enthalpy is increasing and the entropy is also increasing, right? That would be spontaneous for entropy, but not spontaneous for enthalpy. The temperature is a determining factor, right? But this spontaneity is also affected by the chemical equilibrium. So another way of looking at it is if you look at the temperature and the natural log of the chemical equilibrium. So we're going to kind of go back to that equilibrium and pH stuff right now, because the pH can help determine how spontaneous a reaction is or whether or not a protein is going to fold correctly, all based on this equilibrium constant. So just a quick refresher from high school chemistry. The equilibrium constant is the concentration of the products of a reaction. So here I did the dissociation of phosphoric acid. So I have my dihydrogen phosphate ion and my hydronium ions, my products on the top. And the bottom is my reactant, the phosphoric acid. We don't include water because water is a liquid. We only include aqueous states, so dissolved salts, and gases. We never include pure liquids or pure solids in an equilibrium constant. So oftentimes we even leave the H2O off of the dissociation expression because it doesn't actually play into the equilibrium constant. That's just to help balance it and show you where things come from. Okay, so one of the things you need to be able to do is write this dissociation constant. And all you have to do is take the products, the concentration of each product, multiply them together in the numerator, and then you multiply the concentrations of the reactants in the denominator. Okay. So pH is really important in how things function in the human body. So we have biological molecules in our bodies, and the reactions tend to work best at a pH right around 7.4, which conveniently enough is our blood pH. So the thing is, if we get too far off of a pH of 7.4, it can change the shape of proteins, because remember, proteins have this hydrophobic, hydrophilic thing going on. If you change the pH, you're going to change the charges. You're going to make things neutral that used to have a charge, or you're going to get things with charge that used to be neutral. And so the hydrophobic and hydrophilic areas of the protein are going to get messed up, and then that messes up the shape of the protein itself. Okay, pH changes can also change the rate of reaction, because again, it's going to mess with this equilibrium constant a little bit. So biologically, it's really important we stay near a pH of 7.4. So our blood uses a buffer system to keep the pH near 7.4. A buffer from chemistry is just a weak acid with a salt of the conjugate base. in our body, bicarbonate buffers are what we really use, and actually we use that experimentally in the lab as well. It's a fairly common buffer. So we have carbonic acid as our weak acid, and sodium bicarbonate, that's baking soda, as the salts. And what happens is if you add an acid, the acid is going to react with the sodium bicarbonate, and that's going to kind of pull up those free hydrogen ions so that they can't bond to the water. And if you add a base, the base is going to sort of react with the carbonic acid to make more sodium bicarbonate. So either way, it can balance out what's going on. Our body uses a buffer that is based on the carbonic acid buffer, except that carbonic acid actually decomposes to make water and carbon dioxide in a water-based system. So it's actually carbon dioxide that we have instead of the carbonic acid. And so what happens is when you breathe in and out, you're actually helping control the pH of your blood. We'll actually talk about that a little bit more in another lecture. The thing about buffers, and what's important for us when we talk about proteins, is what's going on. How much anion do we have? How much acid do we have? Or in the case of bases, how much ammonia do we have versus the positive ammonium ion? So the Henderson-Hasselbalch equation sort of relates this dissociation, this K that we were talking about in the last slide, to the pH. And it sort of lets us know about the charge of things that are going on. So as we change the pH to pKa, the constant for dissociation, that is a constant. That's not going to change. It will be given to you if you ever need it. The constant's not going to change, but the pH can change. And so this Henderson-Hasselbalch equation sort of tells you, it's set up to keep pH the same. It's set up to figure out what concentration of sodium bicarbonate to what concentration of carbonic acid you need to maintain a certain pH. We're going to sort of turn that over because we're going to look at what happens when the pH balance gets messed up. Because we're going to talk about how that affects proteins, because that's the biggest problem we have as a biological system. And as engineers, that's the biggest thing we need to overcome, is how to keep proteins stable in environments that are changing their pH. So I know from experience with this class that people have a little trouble with the Henderson-Hasselbalch equation and using it. So I want to kind of go through with the carbonic acid buffer how we deal with the Henderson-Hasselbalch equation. Okay, so the first thing we do is we write our dissociation equation. Ka is just the constant, the equilibrium constant for that dissociation reaction. Okay, and again, we have our products, H3O plus and HCO3 minus, and on the bottom we have our reactants, carbonic acid. Okay, so the first thing we do is we just take the negative log of both sides. So on the left side where we just have Ka, we're just going to write pKa for negative log of the dissociation constant. Okay, and then that's equal to the negative log of this fraction that we came up with. So far it's pretty straightforward. Okay, we know that pH is going to be a factor, and pH only refers to this H3O plus. So we're going to do a little math trick, and we're going to take this fraction and we're going to make it the concentration of H3O plus times the fraction that is the anion, HCO3 minus, over the acid, carbonic acid. And so what we do is we take out the pH, and if you remember, when you take the log of two things that are multiplied by each other, you add their logs. So what we get when we pull out the pH is the pKa, that part stayed the same, equals the pH, negative log of the hydronium, minus the log of the fraction. Okay, that's the part that people usually get stuck on. Okay, once we have this, we're going to be interested in the charges, what charge a particular side chain on a protein might have at a different pH. So that way we know if the pH change that we've made is actually going to change what our protein looks like. So we're just going to rearrange this equation. So I'm going to take the log of the anion over the acid, and I'm going to add it to both sides of the equation. And I'm going to take the pKa and subtract it from both sides of the equation. So some simple algebra gets us from here to this rearrangement. Okay. On your homework, you're going to get a little practice with this. you're going to be asked to find out what charge different side chains of a protein have. So we're going to tell you the pH of the system, and we're going to give you the pKa for all the different side chains. And then we're going to ask you is what charge does it have? So if you subtract the pH minus the pKa, you're going to get a number. It's either going to be positive or negative. It's not going to be 0. Okay? So if that number is positive, so actually, I'm sorry, I should say greater than 0, not greater than 1. But I'll change the slide. But it is, if the pH minus pKa is greater than 0, if it's a positive number, then the numerator charge is favored. So in this case, the numerator's charge is negative. Okay? If it's less than 0, if it's a negative number, then the charge of the denominator is going to be favored. There is no charge here, so the charge is neutral or 0. Okay, just a note for those of you who are not very good at dissociation equations and haven't had the practice with it in a chemistry class. When you have the ammonia part of the amino acid, so if you have an NH3 or an NH4 group, it kind of gets flipped over. The neutral is the anion, and the more hydrogen form that ends up on the bottom of the fraction, the ammonia, actually has a plus charge. So you want to be very careful about that when you do your homework. I kind of expect to have some questions about this when we come back to class. That's why your homework isn't going to be due until Monday night after the next class instead of being due on Wednesday. So we have a couple of different biological polymers. The four big classes are lipids, carbohydrates, nucleic acids, and proteins. Okay, so lipids, those are our fats. They're made up of large aliphatic chains with a terminal hydroxyl group. It means it's a bunch of hydrocarbons. It's carbons attached to each other, usually in straight chains, and they have an OH group at the end. Okay. There's also a glycerol, which is just 3 carbons, and each of the 3 carbons has an OH or hydroxyl group on it. And what happens is the hydroxyl groups undergo a dehydration synthesis, so you end up with a glycerol that has 3 large carbon chains attached to it. Sometimes we add phosphate groups, and those are our phospholipids. Okay, carbohydrates, those are just your carbon, hydrogen, oxygen, so we're talking about sugars and starches. So if you have monomers, and most of the dimers are actually cyclic alcohols, so there are hydroxyl groups, but the molecules form a circle, so they're sugars. But the sugars tend to be in their circular forms. So usually a 5-carbon ring with the 6th carbon sticking off the side chain. Okay, there are some straight chain alcohols that are carbohydrates as well. They're just not as common biologically speaking. The next major class is the nucleic acids. And nucleic acids have a carboxylic group. It's a weak acid, but you basically have that COOH on one end. Okay, you have a nitrogenous base, that NH3 or NH4 area, and then you have the monosaccharide and phosphate backbone. Okay, so you have a backbone of the sugars, whether it's ribose or deoxyribose. Okay, that has the carboxylic groups on it. And then you have the nitrogenous bases. Those are your A, C, G, and T that you've talked about in biology a million times, I'm sure, and we will talk about that. We're going to talk a lot more about DNA and RNA next week. Okay. Monomers, so those are the simple sugars or a single base in a DNA or RNA chain. They're joined by a hydrolysis reaction. That means that you have water being added to break something apart, or you have a dehydration synthesis. Most sugars are disaccharides. That's our quick energy. Most lipids are triglycerides. That's used mainly for energy storage, but it also can be used structurally, like in the cell membrane. An important note, most lipids are triglycerides, but our body treats diglycerides exactly like triglycerides, no matter what the FDA tells you. Most nucleic acids and proteins are really big. They're not just 1 or 2 chains added together. Usually they number in the hundreds. Okay. Proteins are really important, so we're going to spend a lot of time here talking about proteins. One of the things you need to know about proteins are they're absolutely the most diverse and complex macromolecule in the cell. In fact, originally people were sure that proteins were what caused heredity because there are just so many different proteins and they're so varied. They were sure that that was the answer to what we now call genetics. It actually took a while for us to recognize that it was actually the nucleic acids. Proteins are used for pretty much everything. They catalyze reactions. They're used for structure. They are, most of your muscle tissue is protein. There is a slight amount of information storage, but the biggest class is probably messengers and catalysts. So proteins are used for a lot of things within the body. Okay, so it's really important to understand proteins. Proteins are made up of amino acid residues. There are about 20 left-handed amino acids that are used in making up the majority of proteins in our body. And they're folded up, so instead of being just a straight line of all these amino acids, they form these structures, right? And there are usually active regions, so there's parts of the protein that not much is going on, and then a pocket or something that is the active part. It's a part of the protein that has to be working. So there can be some slight changes away from the active region, and it won't affect how a protein works, but the active site is what's really important. Okay, proteins have 4 levels of structure, and those 4 levels of structure are determined based on entropy effects, and the hydrophobic and hydrophilic amino acids are going to control that. So if you have a lot of hydrophobic bases, it's going to fold together and not give a lot of surface area to the water. If you have a lot of hydrophilic bases, a lot of charged amino acids, then you're going to have a much more open protein that's a little flatter, and it's going to give more surface area to the water in your body. The thing about proteins is that they're both pH and temperature dependent in their folding. So if you have a very high temperature or a very low temperature, they're not going to fold correctly. Also, if the pH changes, you're going to change the charges of those amino acids. So you're going to change how many are hydrophobic and hydrophilic. Okay, so it's very dependent on pH and temperature. An important truism is protein structure equals protein function. So if you know what a protein looks like, you know what it does. So this is a sort of common thing used by geneticists and bioinformatics. If you find a protein or a string of amino acids that are the same in a bacteria and a human, they're going to do the same thing. If you get the same picture of a protein, the same sort of 3D structure, it's going to have the same job. It's much more dependent on the shape than on the actual amino acids. And at the same time, if you know what the function of something is, you can take a pretty good guess at what the protein looks like. Okay, so what are the levels of structure? The first and simplest is the primary sequence, or the amino acid sequence. Okay, this is literally what order do the amino acids come in. This is the easiest one to figure out, and your body needs nothing more than this amino acid sequence to make a perfectly folded protein. Unfortunately, we are not able to replicate this in the lab all that well. There are too many other things that go on. Your body has chaperone proteins and a bunch of other things to sort of help them fold correctly. Even using computer modeling, we have to pre-fold certain sections mathematically before we can actually get a protein to fold correctly. Even now, although we are getting better and better at coming up with ways of predicting the structure of a protein based on some amino acid sequence. Okay, the secondary sequence, or secondary structure, this is sort of the beginning of what we would normally think of as structure. So we have 4 sort of things that form in secondary structure. The first are these alpha helixes. So if you look at like these little red twisted areas, okay, that's formed mainly by hydrogen bonding between the amino acids to form these sort of helix structures. Okay. We also have beta sheets. These are those flat, broad yellow areas. Okay. Beta sheets are another secondary function. They are also hydrogen bonded together. It's where you sort of have layers of protein, layers of amino acids sort of over each other. Okay, we also have loops and turns. So loops are these green sort of floppy areas. It's basically a string of amino acids that don't form a helix, they don't form a loop, but they give some flexibility, some freedom to the protein. So you see these a lot in proteins that have to change their shape in order to function. So a good example might be a protein that is a membrane channel. That has to change its shape in order to function. So you want some freedom for it to go from one form to the other. Okay, you also have turns. Those are sort of short strings that are connections. So you can kind of see one here. You have an alpha helix that's going into a beta sheet. So you have sort of a little segment of DNA that connects those to each other. Okay. This is the part that we sort of have to pre-fold on our current programs that are trying to determine shapes. So we might tell it that this section is going to form an alpha helix and this section is going to form a beta sheet, and then let the computer program sort of fold it from there. Okay, we also have tertiary structure. For smaller proteins, tertiary structure is what we have. You get sort of domains. If all you have is a small protein, then the tertiary structure is also the active protein, and it's going to have an active site. So this protein is a good example of something where the tertiary structure includes the active site. Okay, but some of our bigger proteins also have what's called quaternary structure. So we have a bunch of these tertiary domains that are subunits, and the subunits will come together to form an active site for an enzyme. A good example of a larger protein that has a quaternary structure is hemoglobin. Hemoglobin actually has 4 subunits that come together. Each subunit is folded by itself, and then the 4 come together to form the active site that is holding onto the iron molecule, or sorry, iron ion, that can then either attach oxygen or release oxygen. So again, the nucleic acid sequence, the primary structure, actually determines the 3D structure. In our cells, we have molecular chaperones, which help. Nascent protein is just the protein as it's growing. This is not a fully formed protein. The protein synthesis happens on the ribosomes within the cell. And what happens is as the amino acid chain grows, some molecular chaperones will come along and attach. It stops things from folding too early. If something's supposed to form a beta sheet with another section of the protein that's going to come later, you don't want it starting to form an alpha helix right away. So these chaperones kind of help the protein, the amino acid sequence, from folding too early to make the wrong type of secondary structure. And so it helps it then later form the tertiary structures as well. Okay, the secondary structures are held together by hydrogen bonding. And there's actually one special amino acid, cysteine, that's really important for stabilizing protein structures, especially ones outside the cell. So cysteine actually has an SOH group, so it has sulfur on there. And what happens is if you have 2 cysteine molecules that are sort of near each other in the tertiary structure, it doesn't really happen in the secondary structure. It's only when you start having these loops and turns and alpha helixes and beta sheets that have cysteine that fold near each other. What will happen is they will form a disulfide bond or a disulfide bridge. So it might attach an alpha helix to a beta sheet that's way further down to help it bend to form the right structure. People are actually fairly familiar with how these work. A good example of the disulfide bonds that cysteine can form stabilizing a structure is actually your hair. Your hair is made of a protein that has a lot of cysteine in it. So if you're like me and you have annoyingly pin-straight hair that you can do absolutely nothing with, I get something called a perm, where I put a bunch of smelly chemicals whose job is to break the disulfide bonds in my hair, curl it up tight, let it sit, and give it time for new disulfide bonds to form with my hair curled up. And then when I take the curlers out, the new disulfide bonds have formed, and the hair theoretically is going to stay curly. In my case, the best I can get is a little bit of body, but other people are luckier and can get actual curls. Interestingly, there are also people who have annoyingly tight curls that they want to get rid of so that they have some straightness so that they can do things like braid their hair. So some people get a perm to get the curls out of their hair and get it straighter. It's the same chemical, but in the tightly curled hair, there are more of the disulfide bonds, so you have more of them to break, so you need more of the chemical. And so they actually, instead of giving you more of the chemical, they make it a much more concentrated chemical. And so if you have curly hair that you're trying to straighten with a perm, you're much more likely to have to do it more frequently and also to end up with burns on the skin of your skull because it's a stronger formula. But it's the same idea. You put the chemical on, it breaks all the disulfide bonds, and then you straighten the hair out and hold it in a straight position until the bonds reform and you have straighter hair. Let's see. So protein function is related to its structure. I think I've said that enough times for you to know it's important. It is both the tertiary and the quaternary structure that define an enzyme's active site. So in a small protein, it's a tertiary structure. If there is quaternary structure, then the quaternary structure is going to define the active site. Okay, but the secondary structures are also important because you need the secondary structures to form the tertiary structures. So those beta sheets and alpha helixes, those are determined by the polarity. So if things are polar, then the polar amino acids are going to try and face the outside of the protein, and the non-polar parts are going to try and face the inside of the protein and sort of bond to each other. So that's that whole hydrophobic, hydrophilic. And then hydrogen bonds are going to help stabilize that. They're going to form between two molecules that are on different parts of the secondary structure. Okay, this is the part where the pH changes actually make a difference. So if you change the pH, you're going to change the charge of an amino acid. You might take something that was negatively charged and make it neutral. Well, now it's no longer polar, which means it's no longer hydrophilic. So now it's going to want to bury itself instead of being on the outside. If you have enough of those changes, you can take an alpha helix and turn it into a beta sheet. Or you could take an alpha helix and turn it into just a turn or a loop. And that change in the secondary structure automatically messes up the tertiary structure, which is holding those secondary structures together into a globular protein. Okay? And so what happens is, if you change the pH, you change the shape of the whole protein. If the protein's shape, structure changes, its function changes as well. You can make it completely inactive. Okay? So proteins that have similar shapes have similar functions. So if we know the sequence of a protein, we can use what we call sequence homology analysis. If you have 70 amino acids in a row that are very close to each other, maybe one or two changes in there, especially if the change is hydrophilic for a hydrophilic, then you know what the new protein is going to look like, and so you can take a pretty good guess at what it's going to do. Okay. We have a lot of different types of proteins. Enzymes are probably the most important. They catalyze reactions, and in particular, they catalyze ATP to ADP. Pretty much every reaction that goes on in your body doesn't happen spontaneously at a temperature as low as our body temperature, so we have proteins that are catalyzing the reaction. Proteins are also important for structure: collagen, elastin, keratin, all the things that sort of hold the pieces of our body together and hold our skin together tend to be proteins. Proteins are important for motility, the actin and the myosin in your muscles. Tubulin is also an important protein for motility. Regulatory proteins, they're going to bind to DNA to switch things on and off. Storage, ovalbumin is the protein in eggs. It gives energy to a fertilized, pretty much any vertebrate. A fertilized embryo is going to be living off of ovalbumin or albumin. Casein's another storage protein. Okay, hormonal proteins like insulin, nerve growth factor, those are going to dictate what's going on in your body. Those are messengers for your body, and they regulate your metabolism. Proteins are also the receptors. So if you want insulin to work, your body has to recognize that insulin has been produced, and the cells in your body have to react to that. So the receptors that recognize that are also proteins. And I think I already mentioned that proteins are used in transport, so they can be used to carry small molecules or ions across a membrane. Okay, the ones that I've listed in blue are some of the types of proteins that we're going to study in this class. They're very relevant for engineers. The ones that are in black are really only relevant for biologists and biochemists. And then the special purpose proteins, that's in green. That's used by different people for different reasons. So depending on your research, it may be important to you. A good example, this is green fluorescent protein. We use that a lot of times to mark things. So if a reaction happens, it will make the green fluorescent protein glow. Okay. Proteins in your body often have post-translational modifications. So every person and most animals make all the same proteins, but they do slightly different things. And every cell in your body makes the same proteins, but not every cell in your body does the same thing with them. So there are certain proteins that the gene regulatory proteins are just not even going to have them get made. We're just never going to transcribe that DNA in some of your cells. But other proteins are made by every cell in the body, but it's used by one type of cell and it's excreted by another type of cell. And so what happens is your body adds things to the protein before it secretes it out into the bloodstream. So some of the more common ones, a bunch of them are listed in table 4.1 in your book. But some of the more common ones are things like glycosylation, which basically glycose, sugar. It's basically adding sugar pieces to asparagine, serine, or threonine. It kind of helps if you have a protein that's made in one cell and it's going to be secreted to be used by other cells. It helps the protein to keep its shape if you have these sugar molecules on it. It also helps your body in recognition that it belongs to you. Okay, phosphorylation is another common one. And this helps to improve the stability of a protein. So if there's a protein that you don't want getting denatured, proteins that are made in normal cells in your body, they're going to travel through your bloodstream at a pH of 7.4 and then end up being used in your stomach for digestion, where the pH is significantly different. They're going to have phosphorylation to help stabilize them as they go through these different environments. Okay, myrostylation, that's adding a really long chain. It basically makes it an anchor. So we tend to have this with things that are involved in our immune response. You want to anchor the protein to your cell, but you want the protein itself sticking out into the interstitial spaces. So you have the protein, and then on the N terminal, so the nitrogen ends, the N-terminal glycine, which is what every protein starts with anyway, you end up adding this really long hydrophobic chain that sort of gets embedded in the cell membrane, and it tethers the protein to the cell. Okay, the last thing I want to talk to you guys about is enzymes, which are one of the major classes of proteins that we're going to deal with as bioengineers. Their main function is to lower the activation energy of important reactions so that they can happen at temperatures as low as in your body. And the active site for an enzyme is specific to its intended substrate. A lot of times we talk about something called a lock and key model. In a lock and key model, there's only one thing that can possibly fit. So if you're supposed to be an insulin receptor, the only thing that fits in there is insulin. That's not a good model. It's a good picture. It's easy to understand where only one thing fits, but it's not realistic and it doesn't match what we notice. If you think about it, pain medications, insulin blockers, beta blockers that are used to help people who are in pain, some of the MAOI inhibitors that are used, those are molecules that look enough like the intended substrate to fit in the spot and take up space, but aren't going to cause the reaction. So they basically stop things from binding to their receptors. So that's a pretty good evidence that the lock and key model is not realistic. A more realistic model is the induced fit model. which is that you have an enzyme, you have a substrate. So if we use insulin as an example, you have an insulin receptor on your cells, and you have insulin floating around in the bloodstream. And when the insulin gets close to one of these receptors, it changes its shape a little bit. The enzyme changes, the receptor changes its shape a little bit. And then if they're close enough of a fit, they'll change a little bit more and unfold each other until they fit enough to make the reaction happen. So in an induced fit model, you can have a lot of different substrates that will all fit on the same enzyme. And that's more how we think things work in our body at this point. Okay. The main thing enzymes do to work to lower the activation energy is that they stabilize transition states. So the transition state is that really high energy halfway between what you started with and what you end up with. It's the reason that reaction diagrams have a hill. So if you can stabilize that state, you can make the reaction happen a lot faster or at a lower temperature. So for example, if you have to break a bond, you might have a protein that is an enzyme, and you have whatever molecule you need to break the bond, like for instance ATP, can attach to this protein, and the protein changes its shape, the enzyme changes its shape and puts pressure on the bond for ATP to break off the 1-phosphate and release the energy. So when you put that pressure there, it forces the ATP molecule into a slightly different shape, and it makes it easier to break that bond. Okay? An important thing, an enzyme is never going to affect the Gibbs energy change. If a reaction is spontaneous, it is spontaneous with the enzyme or without the enzyme. If a reaction is not spontaneous, adding an enzyme does not make it spontaneous. Gibbs energy change is a state reaction. It only matters what the starting point and the ending point are. It doesn't matter how you get there. So an enzyme can never affect the overall Gibbs energy change. It cannot affect the overall spontaneity. What it can do is help a reaction that's going to happen anyway happen with less energy by stabilizing the intermediate state. Okay, the last thing to know about enzymes is sort of enzyme kinetics. So how well is an enzyme going to work with a particular substrate? So the Michaelis-Menten equation is what we usually use to govern the reaction kinetics. So it's basically the rate of the reaction is equal to the maximum rate of reaction. So if you had an infinite amount of the substrate, how fast would the reaction go? Okay, and then you multiply that times the substrate concentration, how much substrate you actually have. Okay, and that's divided by the substrate concentration plus the enzyme substrate affinity, the Km, the Michaelis-Menten affinity. So this enzyme substrate affinity tells you how well the substrate is going to stick to the enzyme itself. Okay, so the Km is on the bottom. So if you have a higher Km, you're going to have a lower rate of reaction. Okay, so that's important to note. Okay, so the enzyme substrate affinity, it's how well the enzyme and the substrate stick to each other. Are they going to pop off immediately, or are they going to stay glued together? Okay, so you have some homework which comes out of the textbook, and on the Blackboard site, the questions themselves will be posted in addition to where to find them. So in chapter 4 of the textbook, I want you to do problems 1, 6, and 8. Problem 1 is kind of tricky. That's the one that involves the Henderson-Hasselbalch equation. So remember to check all of the ionizable groups. It's okay to Google the peptide. There's a tripeptide that we're asking you to draw a picture of. It's okay to Google it and cut and paste that picture into your Word document that you post your answer with. I want you to see what the tripeptide is. You don't need to be able to draw it yourself from scratch. Okay. What is important is that you're able to use and interpret the Henderson-Hasselbalch equation correctly. Problems 6 and 8 are pretty straightforward. They shouldn't take more than 2 or 3 sentences for each part, but you want to make sure you answer the why part of the question. Don't just tell me which of the proteins has the highest affinity, explain why. And if you have any questions, you can email me or you can ask me before or after class next Monday. Hope you guys enjoy your snow day.